welcome everyone today. We're so excited because this is the first session that we will have from this WeCancer Seminar series. We are CERFA. This is a society of German researchers Spanish researchers in Germany. And this seminar series has been organized by a subgroup within CERFA, which is CERFA Cáncer. So we are mostly researchers interested in cancer research. And this today is the first of, we hope, many series of seminars in which we want to share together some of the new studies in research, in cancer research. And we are still open to receive more candidates. So I think, Aithea, you shared already the link through the chat. So you can email us if you are interested in being one of the speakers and share your research with us, and we can have some nice scientific discussion together. So Jose, I think now for you. I will stop.
Hi everybody. I am supposed to not present Elisa. Juan was going to do that for my side.
Okay,
so John, I give you the.
Let's go. So actually, I almost asked for this pleasure to introduce Elisa, who before coming here to Germany, I didn't know, and now she's become one of my favorite persons in the world. So I'm really happy to be here to introduce her. Elisa made her PhD in the IRB in Barcelona. She was very successful there. She managed to establish a new line for Eduard Bachet's lab, where she pictured TGF-beta as one of the main drivers for metastasis in colorectal cancer. And then she moved to here to Heidelberg, to the DKFZ, to continue with a sort of a similar project in which she was characterizing the different cell types in the pancreatic tumor, and she will actually expand that a little bit more. She has had a quite successful postdoctoral period, and now it's starting to come out on the world to be published, and she is now preparing the next step where she hopefully will become an independent researcher, and I really hope so, because I would love to collaborate with her for a really long time. So I leave the stage to her and prepare to be amazed with some nice data.
Thank you, John or Juan, for the nice introduction. I will share my screen now. Let's hope it works as before. Okay, there we go. I will also do this smaller. Oh, okay. See you all there. Okay, so this is the title I chose for the talk. So I'm very honored to be the first one to talk. I hope that this will work nicely and that we can have many more talks, and also eventually invited speakers also from outside the CERFA group, so that we can invite people. Actually, some people told me they would love to show their projects, some group leaders from Spain, so this would be really nice. I think people will be interested in this. So this is the title I chose for my talk. It's quite long. I will read it once and then I will split it. Aggressive pancreatic cancer shows hypomethylation of repetitive elements and the execution of an intrinsic interferon program linked to a ductal cell of origin. So more than a title, is almost an abstract, because basically it's telling you everything that I will show you. So just to make it a bit, no, it's not working. Arrows, let me see. Okay, okay. So just to split it a bit, that it's not so complicated, today I will be talking to you about pancreatic cancer, about methylation, and specifically hypomethylation of repetitive elements, also interferon signaling, as maybe mentioned, John mentioned already, and cell of origin. So if you are interested in one of these, I hope you will find the talk interesting. I'm very happy that this project or this work has now recently been accepted or published in Cancer Discovery. So I will be today mainly showing you main figure data and some supplementary data, but as for any project, you know how it doesn't go, that's how the reality is. So there's of course always much more in the back, much more behind that we all do. So I hope we can do it very like open today. And if you have questions, hopefully with some of the actual data, extra data that is in the back, I'm able to answer your questions and we can have a nice discussion after the presentation. So when we talk about pancreatic cancer or pancreatic ductal adenocarcinoma, PDAC, which is the biggest type of pancreatic cancer, so I will be using either pancreatic cancer or PDAC, pancreatic ductal adenocarcinoma during the talk. I think it's for us all clear that it's a very bad disease, and it's actually exemplified in this slide here. So it's the type of cancer where actually new cases almost equal the deaths every year, and this actually translates into pancreatic cancer being the type of cancer with the lowest survival rate. That it ranges from 7% to 10% depending on the country where you look. And as you can see also when you look to the pink bar, this is actually the change in survival in the last years compared to other tumor entities. The change in survival that we are able to reach in pancreatic cancer is actually not much. It's also actually another way to see it here. This is like how the survival changes along the years. And as you see, pancreatic cancer always is the lowest, and even though there is a bit of increase now, especially in the last years, this is not comparable to what we can achieve or we have achieved with other tumor entities. And it's indeed the case that pancreatic cancer is estimated to be the second leading cause of cancer death in 2030 in US, or the third in 2025 in Europe. And this means that we still need to do more and to learn more about pancreatic cancer. And many promising drugs that were arising always from the bench sites or from mouse models or cellular models unfortunately failed into the clinic. And it's only since very few last years that we have some new therapy options, but these actually just imply a minimal improvement in the patient survival. And actually the majority of the treatments remain mainly palliative. So again, this means that we need to do more, and it's actually in the last years, there's a lot being done and we are learning a lot, I think, but there's still, of course, much more need to improve this situation. So when we talk about pancreatic cancer or human pancreatic cancer, this is what we are talking about. So this is just a section of a tumor from a patient that went resection here or surgery here in Heidelberg. And as you can see, pancreatic cancer is actually formed by very few epithelial cells that form these glands you can see here. And they are embedded in this massive amount of stroma that is mainly composed by so-called activated stellate cells. So this is basically CAS or cancer-associated fibroblasts. Few immune cells there and few endothelial cells. And this, when you see the picture like this, it's just to illustrate that actually when you analyze human samples, it gets very tricky to indeed understand what's happening in each of these cellular populations because they are all intermingled. So when analyzing bulk tumor data, it's very hard to understand which signal is coming from where. But what is clear, I think, for all of us working in cancer, or also people not working in cancer nowadays, is that the presence of these cells is just like not passive, but it's really making a difference. Like each cell is influencing the other, and there is definitely a big crosstalk between all these different cellular types existing in the tumor. And this different crosstalk is going to influence, of course, the fate of each of the cell types, so also of the tumor epithelial cells, and ultimately the fate of the tumor and the prognosis of the patient. So it is important to understand the cells inside, in situ, in the tumors and what's happening in there. Now for pancreatic cancer, this is quite technically complicated because actually few patients undergo surgery, so it's quite hard to get primary material from patients. Now there are other technicalities that make it complicated, such as, for example, pancreatic cancer. Pancreas is actually the source of RNases, sorry. So when you want to do transcriptomics, for example, it makes a bit complicated. But nevertheless, some years ago, we decided to actually start our research from this as close as possible to this situation. So basically studying primary human material to start from that point. And very luckily, we could establish a nice collaboration with the surgery department here in the University Hospital in Heidelberg. And from collaboration with them, we obtained primary materials of primary pancreatic cancer samples, not only this, but also actually distal normal pancreas to be able to compare from patients undergoing surgery here in Heidelberg. So we established a protocol from these fresh samples, specifically isolate the epithelial tumor cells to actually understand specifically what's happening in the tumor epithelial cells. And what we are having a look today here, what I will show you today, is that we isolated these epithelial cells and we derived a whole genome transcriptome by RNA sequencing and the whole genome methylome, so DNA methylome by bulk whole genome bisulfite sequencing. So when we first look at the DNA methylome of this specific tumor and normal epithelial cells, we of course found many differences. We found many so-called DMRs of differentially methylated regions. So there are many regions in the DNA of this tumor and normal epithelial cells that change methylation are very different. I'm not going to go into detail there today. There are many interesting candidates, but I don't want to show this too much today, because something that was very surprisingly for us, and this is what I want to start now the data with, is that what we found when looking at these regions of the genome of the DNA that are differentially methylated between normal and tumor epithelial cells is that, of course, the normal and the tumor epithelial cells, they cluster apart very differently. That's what we are looking at. But now, very surprisingly, the tumor epithelial cells, or the different samples, tumor epithelial cells, they cluster into two different groups. And now you have to remember always this is based on DNA methylation profile. So we didn't know at that point what this meant. So we decided just to call the two clusters methylation cluster 1 and methylation cluster 2 without having more information on this. And because we did have also the transcriptome of these two groups of these samples, we basically went to the very simple question: are there any differences in transcription between these two groups? Do they express different genes, these two groups, based on methylation? This is just to validate that the groups are robust in methylation. But going back to the transcriptome, we asked this question: are the different genes expressed between the methylation cluster 1 and 2? And definitely, yes, there were some different differentially expressed genes, and very clearly the top ranking signatures that came upregulated in one of the methylation cluster 2, was interferon response related signatures. We built an interferon signature that we use to differentiate the two clusters, and part of the signature is, for example, the transcription factor STAT1 that we believe is one of the master regulators of the whole process, and that you can see here we could actually validate at the protein level, specifically express higher in the epithelial cells of the methylation cluster 2 samples. So this meant, sometimes it's stuck. Sorry. So this meant that somehow, based on methylation profile, we were able to separate epithelial tumor cells that are interferon high or STAT high versus those that are interferon low or STAT low. So what we next did, of course, is to check how about interferon signaling and pancreatic cancer, because back then there was no much. And what we clearly found when analyzing public data of pancreatic cancer, for which there is a clinical annotation, is that those pancreatic cancer patients with higher expression levels of STAT1 or interferon signaling, they clearly are associated to worse outcome compared to those with lower levels of interferon signaling or STAT1. For those in the field, maybe none of you, or but some of you are in the field of pancreatic cancer, there are some subtypes as for other tumor entities described for pancreatic cancer. Mainly there are two of them, the squamous and the pancreatic progenitor. And the squamous is more associated to more aggressive tumors, and actually nicely fitting with our wall survival observed here. The squamous or tumors assigned as squamous type, they actually also express higher levels of the interferon signature or the STAT1. So we do find certain correlation between these, but it's not exactly the same, as we can actually see that if we take the supposed to be better survival patients, we can further separate them in low survival or higher survival based on expression of STAT1 or interferon signaling, indicating that it's complementary to the previous annotations. So we then next wonder, okay, what are the differences between these two clusters? What is actually driving these differences in interferon signaling itself? And the easiest explanation, of course, would be, and the project would have finished much earlier, but less interesting also. The easiest explanation would be like, well, there are differences in methylation of the interferon genes themselves. And this is basically one of the groups, why one of the groups expresses higher levels of these genes. So what we did and is actually to analyze differences in methylation now between the two tumor groups. And actually there are quite some differentially methylated regions, or DMRs, more than 3,000. That's quite a lot, especially if you consider there is not that many samples. However, we could not see differences or any correlation to any interferon-related gene or process in these regions that are differentially methylated. None of these genes correlate, none of these changes in methylation correlated to changes in expression to any gene on the genome. So somehow there are big differences between these two tumor groups, but these are not directly influencing gene expression itself. So there might be something else that is driving this, or there might be something else different between these two groups. But what is this? What is actually different between the methylation cluster 1 or interferon low and the methylation cluster 2 or interferon high group? And this took a while, and I must say it was a bit by chance when looking to some QC plots from the bioinformatician I was working with. That we found one day, I actually found this plot. So here you see basically the density of methylation along the genome, so in all the DNA of the samples, and each column represents one of the samples of the tumor samples that we have. And you don't need to understand it so really in detail, but I think it's really clear to see that these 3 samples here are very different to those 4 samples here. These actually having darker color here implicated higher methylation level or more density of methylation, so more methylation along the genome in those samples. And very nicely, when I asked to the bioinformatician which sample is which sample, this is what it came out. And what we clearly saw now is that these 3 samples that show overall in the genome lower methylation level corresponded to the MC2 or interferon signally high tumors. Now, when I say that there is this global lower methylation levels along the genome, it's partially true, because it is true that overall you have less methylation, but actually now when we look at specific genomic regions, it's not that clear. Specifically when we look at CPE island sites, it's now not longer that clear that the interferon high or MC2 tumors are lower methylation. Here the profile is a bit lost. Indeed, if we try to classify our tumors based on methylation profile only in CPE island sites, we cannot pull apart the tumors, so they're very different to the normal, but between them we cannot see differences. What we found is that where the differences lay is actually in regions of the genome that contain repeated DNA sequences or repetitive elements, named line, sign, and interestingly LTRs, long terminal repeats, or endogenous retroviruses sequences. It's in these regions of the genome where the methylation cluster 2 or interferon high have a marked loss of lower methylation compared to the interferon high tumor epithelial cells. And why this was so exciting when we found this is because some years back there was several papers. Now there have been others, but they were the starting ones in 2015. What they did is actually they wanted to study the mechanism of action of DNA demethylated agents. So when you globally demethylate the DNA with the chemical compounds in the clinic, how this actually controls the decrease in proliferation or apoptosis induced in the cells. And what these papers found is that actually when they impose this global hypomethylation to the cells now de-repress long terminal repeats or endogenous retroviral sequences that normally are very methylated, so they are not expressed. Now without methylation, they start to be expressed. And now some of these ERV sequences or endogenous retroviral sequences can form double-stranded RNA by complementary pairing. And now this basically is sensed by the cell as a viral infection. It's just that it's not external RNA that comes into the cell, but it's the same RNA from the cell that should be silent by methylation, but now starts to be expressed. And this so-called viral mimicry, this response to this double-stranded RNA activates interferon signaling and finally has the action in the cell. So we thought maybe, even though actually our tumor cells are very happy, and we can discuss later why this could be, maybe actually this is something that could be happening in our system. And this is why the ones that are lower methylation have higher interferon signaling levels. And we could actually have a look at this. What we did is to go back in the RNA sequencing expression of our two groups of tumors, and we looked for endogenous retroviral derived sequences. And we could see that, indeed methylation cluster 2 or interferon high tumor cells, they do have higher expression of repetitive elements, so line, and especially more stronger ERVs. Furthermore, they also have a high expression of a response to double-stranded RNA that was actually very nicely correlated to the extent of expression of interferon signaling. And also we could see that expression of certain repeats correlates with this extent of activation of interferon signaling. This is just to show that we could validate this in a larger cohort of patients, so you don't need to go much into the details, but you see again, we do see two clusters based on methylation profile, and the orange ones here, they have this lower methylation of repetitive elements or genome-wide, but not at CPG island sites. And again, we see this upregulation of interferon signaling response genes or repeats in these lower methylation tumors. So this is a validation set of 11 additional tumors. We next moved in vitro to see if we could find some, we could have some tools in vitro with which we could play around and understand a bit more what was going on. So what we did is go into a panel of primary derived, so primary cell lines, pancreatic cancer cell lines derived from patients. So here it's 7, it's actually always 7 the number, now I just realized, 7 different cell lines you can see in columns, and here we are looking at the expression of interferon related genes, and as I think it's very clear to see these 3 cell lines express much higher levels of interferon related genes compared to the other 4 cell lines, and this includes, of course, our favorite always, that one. And of course the signatures are enriched in these cell lines, and now very nicely fitting with our analysis of the patient data. When we put these interferon high and interferon low tumors into mice, those mice bearing tumors from interferon signaling high tumor cells, they survive less or have shorter survival than the other one. So always pointing to this more aggressive phenotype of these interferon signaling high tumor cells. We performed methylation, of course, of these cells to see if we could also validate that these interferon high cells are less methylated than the interferon low, and this was actually the case. You can see here that specifically at repetitive element, genomic losses containing repetitive elements, we do see these lower methylation levels in the interferon high tumor cells. And now nicely, we take those tumor cells that have higher methylation level and we treat with the methylation, we bring them back to this situation, as you can see here. So now the green cells, basically, that are interferon low, they start to express STAT1 when we impose demethylation. Not only this, but finally, it was very nice to see that indeed these interferon signaling high tumor cells that we had in vitro, they did have higher levels of double-stranded RNA in the cytoplasm. And at the expression level, they also express higher levels of these double-stranded RNA sensing elements that again correlates nicely with the extent of activation of the interferon signaling, indicating that what we saw in the patients it can be actually regulated intrinsically by this activation of this double-stranded RNA in the tumor cells. Finally, to validate or to prove this, we perform knockdown of MAFS, is a master regulator of the double-stranded RNA sensing machinery. And as you see, when we downregulate the capacity of the cells to sense double-stranded RNA, now the expression of interferon-related genes also goes down. Indicated that actually it is the case that sensing this double-stranded RNA present in the cells is at least one of the reasons why these cells have higher activation of these interferon-related genes. Now, if interferon signaling or response to double-stranded RNA is linked in our hands, what we see, and in the patient data towards survival, the next question, of course, was what happened if we inhibit all this interferon signaling. And this is what we did by different means. So here by genetic downregulation of STAT1 as this master regulator of the whole interferon process, or MAPS, this master regulator of the response to double-stranded, or the sensing of double-stranded RNA. But also we did a more close to the clinic approach, meaning pharmacological inhibition of tumors that are already grown in the mice, and now we start to treat with JAK-STAT inhibitors to block interferon signaling pathway. And as you can see here, for all the examples where we impair interferon signaling, the tumors have a harder time to grow. This you can see here, also with the MAPS knockdown, and nicely also partially we can block the growth of the tumor when we, my internet, do you hear me? Because the internet said it's awesome. So also when we give pharmacological inhibition, also we can delay a bit the growth of these tumors. Now, very interestingly, this was true only for the case of the interferon signaling high tumor cells. Nothing happens if we do the same with interferon low signaling cells. And this actually makes sense, because if you never had interferon signaling response, why would you care that it's removed? So it makes sense that the ones having it actually are more susceptible for this. And this, of course, opens this nice possibility that stratification of these patients into these two groups might lead to a good strategy to when going to treat patients with JAK-STAT inhibitors, for example. They might potentially benefit from this. So I want to move now a bit into a more theoretically part, I think, that is cell of origin, that was also in my title. So just very briefly, and you don't need to look at everything, I will just tell to you. So there is this, there are now several studies pointing or showing that the methylation profile that the tumor cells have at the end of the day, so this is basically what you would see here, or sorry, here, it's a mixture of changes in methylations that happen to the tumor cell because of being a tumor cell, so to say. So the tumor cell will change and will acquire differences in methylation, but it might retain some information, some methylation traits from a possible cell of, from the cell of origin, from the normal cell from which this tumor was originated. So basically you have, as I was saying, this mixture of the cancer-specific changes methylation, but you also might carry information from your cell of origin. So you might, when you do, if you have different cell of origins, you will have different contributions in the methylation. And this is an idea that it's very deeply studied in, there are several groups in the DKFZ that are studying this. So this has been mainly studied in blood diseases. So this was especially started by Iñaki Martin Subero, who is now in Barcelona, but also Christoph Plas, who is in the DKFZ, actually worked a lot on this, and they keep on working on this. And now more recently in the group of LICO in the DKFZ, also they started to study this in solid tumors. So of course, having all these people in the DKFZ, we said we have to look at this because we see these profiles of methylation so marked. So again, like it's a bit complicated, but, so for pancreatic cancer, in adult pancreatic cancer, there are basically two types of epithelial cells existing that might be the origin of pancreatic cancer. And these are the acinar cells here in pinkish and the ductal cells here in more blue. Now, while for mouse models, genetic mouse models, both cell types are able, with the right cues, are able to give rise to pancreatic cancer. And as I will also show later, for human is of course debated which is the cell of origin, as it's very difficult to do transgenic human people, as you can imagine. So what we then did, and this was actually quite complicated to get, but we established a very nice collaboration with Epson Arda in NIH, and it's a Romance group in Belgium. You again don't need to look in the whole scheme, but we basically managed to get methylation profile of human adult acinar and ductal cells. And as you can see here, they have a very different methylation profile. This is different ductal samples and acinar samples. This is some mixed samples. You don't need us to focus that much. But it's just to say that we actually managed to have these methylomes from ductal and acinar human cells. So when we then compared to our tumor cells, actually the picture got quite complicated and quite different to what has been shown in blood diseases, where actually there seemed to be quite similarity between normal and tumor samples. That's probably also expected because the blood system is completely different to solid tumors, where you acquire so many mutations and so many changes, right? But what we actually found is that tumor cells from either MC1 or MC2 are completely different to the normal acinar or the normal ductal methylomes. And this is exemplified here. And so basically, if we look at this now methylation, if we look at the methylation of acinar markers or ductal markers, they are basically, this is our tumor samples here in orange and black. And as you see, it's mainly everything yellow. This means all these genes, all these markers are highly methylated in tumors. And this makes sense because these tumor cells are neither acinar, neither ductal. They converted completely in something else. And also, if you just look at all the genes, basically, you can see that the tumor samples cells, as you can see here, and supervised clustering acinar and ductal cells on one hand, and the tumors completely far away on the other one. But basically, this told us that we have to look at the data on another way, because if we actually look to acinar and ductal markers, like acinar and ductal identity as we know it like this, we are never going to find anything because the tumor cells became completely something else. So what we then hypothesized to try to make the picture a bit more clear, and here I have to actually give the credit to John because this idea came from during a lunch break with him. So be nerd and talk science during lunch because it might actually help your project. So thank you, John. So what we reasoned is that if there are some methylation traits that are linked to the cell of origin, specifically now to ductal cell identity, these have to be also maintained in this cell line that it's called HPDE. It's a human pancreatic ductal epithelial cell line. So it's a cell line that we know is established from ductal normal cells. Of course, it went transformation in vitro, but if there is something that is still kept on this ductal identity, it should be also present in these epithelial cells from normal ductal cells. So basically, we had a look to the methylation of these cells in comparison to the normal acinar and ductal, and also as I show you next to our tumors. So here it is to show you this now in blue is this cell line, this ductal normal cell line that is of course transformed via in vitro. But as you can see, actually it has a ductal identity because it clashes with the ductal cells here in, I don't know which color is this, in dark pink. So they actually have this ductal identity. Now when we put together with our tumor samples, very nicely, our tumor samples that are methylation cluster 2 or interferon signaling high, nicely clustered together with this HPDE cell line and the ductal cells, while the methylation cluster 1 or interferon low cluster closer to the acinar samples. And this basically gives this idea or suggests that MC2 or interferon high tumors are more similar or share some ductal origin identity, or are more similar to ductal cells. To explore this a bit more, what we did next is to go into expression data, public expression data from acinar or ductal human cells. And we basically check for the expression of our pathway, interferon signaling pathway that we see differentially expressed in the two tumor groups. And very nicely, what we could see is that actually already in the normal pancreas, ductal cells express higher levels of interferon signaling or interferon-related genes when compared to acinar cells, as you can see here by gene set enrichment analysis, or when we look at specific genes, for example, again, STAT1 is quite much higher in ductal cells compared to acinar cells. Not only this, but nicely, we could see that ductal cells in the normal pancreas already express higher levels of response to double-stranded RNA signature. And not only this, but they already have higher levels of endogenous retroviral sequences. And in general, they are more similar to these MC2 interferon signaling high tumor cells. So all in all, basically, this was now suggesting to us that potentially it is not the same if you acquire a mutation in a ductal cell or in an acinar cell. And presumably, if you acquire a mutation in a ductal cells, you're more likely to go into an interferon signaling high type of tumor, while if you acquire a mutation in an acinar cells, you likely might go to an interferon low tumor type. Now, to actually explore this possibility, I was very lucky to find Janelle Kopp in some meetings. I met her, and she has this nice story where she actually has specific genetic mouse models where she specifically induces mutations for pancreatic cancer to create pancreatic cancer tumors arising from either ductal or acinar cells. So this was actually the perfect model for us, and very nicely again fitting with our correlation of interferon signaling to more aggressive tumors, and also with the patient data. Already she showed in this initial study that those tumors arising from ductal cells, they actually have are worse in the mice than those arising from acinar cells. So this was also really exciting for us to see in her initial publication. Now Janelle has in the last year derived a collection of cell lines from these different tumors. So this is cell lines that we know arise from ductal-derived tumors or from acinar-derived tumors in these genetic mouse models. And it's important to say these genetic mouse models are created with the same exact genetic mutations, because there are other models that are not like this. So what we did is now, or what I asked Janelle is, can you please check interferon signaling in those two? Is it then really true that those tumors arising from ductal cells have higher expression of interferon-related genes? And I guess you expect the answer, because otherwise I would not put this slide, right? So the answer was yes, those tumor cells that arise from ductal cells in the mouse model actually have very clearly higher levels of interferon-related genes. Here just show some examples. Not only these, they also have higher levels of genes responsible of response of double-stranded RNA. So all in all, this leads me to the model, and I just have one extra piece of data to show you. But basically, we envision now these two different scenarios. One where tumor epithelials that we do link to a ductal cell of origin or to an acinar cell of origin. So now those tumors that presumably come from ductal cells, there the tumor cells have this profile of higher expression of interferon-related genes that we show are at least partially mediated by lower methylation of repeat-derived transcript of these repetitive elements that form double-stranded RNA. There might be other mechanisms we haven't explored here. And this situation is linked now to a worse outcome in the patient, or in the mice also, as I show you, compared to a situation where interferon, where tumor cells do not have this activation of interferon-related genes. I've also shown you that we are able to ameliorate the disease in the mice so far if we decrease the activation of interferon signaling in this situation. Now, what I haven't shown you, and I just give you 3 more pieces of data, is some of the reasons why we think that these tumors are actually linked to a worse outcome, or some reasons that can contribute to the worst outcome seen in these patients. One of the things that we clearly saw is that these interferon-high tumor cells are able to activate a complete different program in the surrounding fibroblasts, or stellate cells, so-called stellate cells in the pancreas. This is actually shown here, where you see the transcriptome of normal stellate cells, so fibroblasts from the normal pancreas, so to say. When we put conditioned media from either interferon-high tumor cells or interferon-low tumor cells, or stellate cells as control or fibroblasts as control, as you can see, stellate cells, normal stellate cells receiving the media of interferon signaling high tumor cells acquire a complete different profile to those cells receiving the media of interferon signaling low tumor cells that actually cluster together with the control situation. So even though it's tumor conditioned media, one is completely different to the other one. And this activation here involves interferon signaling itself, but many other pro-inflammatory molecules that we think might promote the recruitment of immune cells that can further influence the worst outcome of the patient. So this different activation that we saw here, it is actually different activation that promotes tumor growth. So now if we take stellate cells that are activated in this way, compared to the ones that are activated in this way, and we put them together with tumor cells to grow tumors, this is what you can see here. The mice that receive tumors with stellate cells activated in this way, in the interferon high, now they grow more than the ones in green here, implicating that the activation of these fibroblasts helps tumor cells to grow more at least in the mice. So this might be one of the ways how these tumor cells benefit from activated stroma, or the stroma gives something back that helps them grow more. Additionally, as I told you, there are many molecules that might promote immune recruitment. We haven't studied this because we are always in immunocompromised mice, but it's something really exciting that I'm actually now starting to do some experiments on that. And yes, to point, actually this interferon is signaling high tumors, tumor epithelial cells, sorry, concomitantly with the activation of these or expression of these in pure interferon-related genes. They also express higher levels of several immune regulatory molecules like PD-L1, for example, or IDO1, and so on and so forth. So probably they, while presumably activating all these pro-inflammatory and more inflammation in the tumor, they might be protected by being attacked by the immune cells. So basically they have a dual way how to be more aggressive. And this was actually it. So this is my final slide. Thank you for staying so late on a Thursday, for listening to me. I'm happy to take questions. I wish you all a happy enter in the 2021. I must say, after all, with the publication, it hasn't been so terrible year for me, even though it's not great, we cannot go home and so on, but at the end, it's what it is. And I think I mentioned most of the people throughout the talk, so you can read all the names there. There have been many people involved in many different branches, and this has been also for me very nice to really touch upon many different things. I think that's the way to go. Thank you. You are all muted, but it's nice to see you clapping in any case.
Okay, let's unmute ourselves.
So thank you very much, Elisa, for your first talk in the WeCancer Seminars. It was a pleasure to attend your fantastic job. Very interesting. For me, that I work in another field, it's very interesting to know how are you working in this type of data. So now it's moment to ask questions. So you can, everyone can write in the text, or maybe raise your hand and ask for yourself if you want. So. Okay, so maybe I can start asking something maybe very basic, because this field is very far from my knowledge. So, because I work in metabolomics, but so
by the way, this is far for me.
Yeah, but so there was something in common that so we use similar statistics, for example, PCA or clustering analysis, but also you usually use. For example, PLSDA analysis to look for, no? Okay.
No, I have to say, like, the bioinformatics was done, most of it was done with two bioinformaticians from DKFZ, two postdocs from DKFZ. So I trust them in doing the thing. So most of it is like all the clustering, for example, was done, but the clustering we did with several methods. So we see it's a very robust thing actually.
Okay, because for example, with the first PCA that you present, so you were able to detect difference between the two groups, but also there was a third group that was difference in the PC1. But so I didn't understand what was the difference between the three groups.
Sorry.
So if
like this. Yes. This is the one you mentioned, right?
Yeah. So the points on the right of the.
Yes, I passed really quickly to this. So because basically here for this PCA that I'm showing, it's not everything. So we are selecting only some specific regions of the DNA. And these are the regions that are differentially methylated between the normal epithelial and the tumor epithelial cells. So basically that's why you see PC1 is 84%, because you basically bias on defining the regions that separate gray versus black tumor cells. So we also did the unsupervised like without pre-selecting the regions, and this works as well. I always like to leave it this way because this is actually how we found it. So like the whole project, I always say the whole project started with was supposed to be the final figure of another manuscript I'm finishing now, where we do more like RNA expression analysis, and then just to have some methylation analysis and compare a normal tumor. So this is how everything started, actually, because we did this PCA. I just wanted to have some figures for supplementary data, you know, to say, oh yeah, and then we have the PCA with the DMRs between normal and tumor, and we will have the two groups, and that's it. And the two groups gave rise to this. But then, of course, we did the unsupervised also to see that it works. Plus, we did the clustering analysis also. So this is just one example, but we use like I don't know, like how many different methods to show that robustly we always see these two groups when looking into the tumors.
Okay, thank you.
But more about informatics, I don't know if you know it.
I have two questions, sorry, but I don't know how to raise the hand in Zoom.
Yeah, go ahead, Maria.
It was super nice talk, Elisa, as we are used always to see yours. And I got super interested, especially with the very last part. So did you look at how the immune infiltrates is different in the two tumors in the patients?
Yes.
And the second would be, could the interferon high signaling patients benefit from checkpoint inhibitors?
Yes, very nice. Yes, I didn't show this, so this is hopefully coming up soon. Because here, right, we use, I mean, in this story, we just talking about the epithelial cells actually, and what we only show here is that there is this higher expression of immune regulatory molecules. So this directly or like suggests that there might be something with the immune system, of course, no. And here we didn't look into this because everything is like immunocompromised mice, apart from the last ones, but we didn't look more into detail with this. So what I do have that I didn't show here, and in the initial slide where I told you like there are all these different tumor types in the tumor. I mean, Maria, you know. But I also did RNA sequencing from the different compartments of the tumor, meaning the epithelial cells, the fibroblasts, and the immune cells of a number of patients, like around 26 patients or so on. So with this data, I can actually look to the reply to your question, Maria. So basically, I can actually interrogate what's the profile of expression in the epithelial cells, and does it actually correlate to some changes in the immune compartment there. Now, the immune compartment I'm checking there is the whole immune compartment, so I'm not specifically looking at T cells, but still I think it's very nice to see. And this is actually the case that you do see these differences. So basically what I see is that, but I think it's also not only the immune system, but also the fibroblasts, as I'm showing you here. So basically what I do see is that if you have higher expression of interferon signaling in the epithelial compartment, you do have higher expression of epithelial signaling in the stromal compartment, like the same patient. And this is not only STAT1 and MX1 and like the classical interferon-related genes, but this is also PDL1, PDL2, actually, either one, and so on and so forth. And this was then for me suggesting that there is this sort of protection, not only from the immune cells, but actually also, not only from the tumor cells, but actually also the fibroblasts, because they all start to express these immune modulatory molecules, or like stopping molecules, the immune system. And now if you look in the immune system associated to this interferon high epithelial and stromal patients, you do actually see higher expression of exhaustion markers in the immune cells. So what I think is happening there is that basically the system, the tumor cells together with the fibroblasts, that driven in the first place, I think, by the tumor cells, are basically pushing the system too much. You have all this activation, you have too much activation of the system in a way that this has been also been discussed by other people. Basically, you overstimulate the immune system probably to the point that it's exhausted and then it's not working anymore. And this is based on gene expression. This is what the data would tell us. One, of course, needs to do some functional validation and models and like this, but this is for correlations in the human data, this is what it suggests. So now for blocking, I mean, the easiest answer is yes, because these patients have higher expression of PD-L1, not only in the tumor cells, but in the fibroblasts. But now there are several studies saying this actually doesn't work, because it's not only PD-L1, you have other things like B2M, for example, has been shown, and this actually, it's already a way how the tumor cells would escape the blocking only by PD-L1. So I don't think it's that easy. And at the end in this paper, we suggest that rather than this, it might be actually a better idea to block interferon signaling, even though it's counterintuitive. But I mean, the thing is, like, there are many papers or several papers now doing this, like acute demethylation or acute interferon treatment or stuff like this. And this, of course, works because then you have an acute activation of the genes, and then you can block PD-L1. No. I think here the situation is a different thing, because it's not an acute, exactly, it's not an acute situation, but it's a chronic situation. So the tumor cells, they learned and they grew and they managed to be in this way. So it's not like you change a cell that never had this and you put it in, no? So I think not the same things that apply to these acute activations apply here, probably. But, yeah, Jose, you were like,
So, thanks a lot. I just want to make a comment to Maria's question. So IDO, PD-L1, and beta-2 microglobulin, they are interferon-regulated genes, mainly interferon gamma and interferon alpha. There has been already published a couple of years ago opposing roles of type 1 interferon signaling in cancer cells and immune cells. So I really think that your data really fit in the human situation very nicely with what others have shown already in murine models, and that's a really cool work. So I have two questions, one regarding CD40 expression on fibroblasts in the interferon high signal or group. Is it increased? And just coming to that because the only immunotherapy who has done something against PDAC was CD40 agonists. But it also killed patients. So yeah, that was one question. Another one goes on the endogenous retroviral elements. Is there a way to cluster or to select certain, are they, so for me it's very difficult to talk about transcriptomics because I am an immunologist, but for instance, do you see a pattern of the same ones being upregulated in all the patients you examined?
Very nice question. So for the first one, I haven't looked specifically for CD40. That's, I mean, all these analyses are ongoing with the fibroblast with the different compartments that's running this week. So I cannot answer to you. I will have a look for sure to specifically CD40 in the fibroblasts. I haven't looked at it. Now for the second question, yes, it's very complicated of the repeats, but it's a very good point, and I think it's very interesting, and I think a lot more will come in the next years because it's really crazy. Now there are also papers with SIGN, and I mean also line and jump into the genome and do other stuff. Now for the ERVs themselves, for the endogenous retroviral sequences themselves, all good. The baby is sleeping.
Yeah, I am getting kicked out of the room.
So for the sequences of the endogenous retroviruses, it is, I think, in one way more complicated, in another way maybe easier also. In the way of looking at the ERV sequences, it's complicated. As I show you, or I mean, I didn't go into detail, but when I show you the expression of the repeats, it's sort of a density thing. So you do have more of some, but not of all of them. So it's definitely not an on-off. So it's not that all ERV sequences get all of a sudden expressed. There are some that are higher expressed and some that are actually equally expressed in both tumors. Even there are some that are more expressed in the other. So probably there is more to come, I think, in the sense of there might be differences, but this I don't know. But there might be some differences on how stable they form double-stranded RNA, for example. I mean, there might be some ERVs that are more prone to jump into the genome and to create problems in this way, some that are more forming double-stranded RNA and going more into the double-stranded RNA sensing machinery and so on and so forth. So we have looked to some specifically that have been previously published to activate this double-stranded RNA response and so forth. And we do see these in our cells and in the patients also enriched. But these sequences appear also multiple times in the genome. So overall, we do have more sequences of a specific ERV that has been linked to this situation, to this double-stranded RNA. But not for all the locations in the genome where this sequence appears. So I think it's much more complicated like this.
Okay.
In the easier explanation, I must say that what we see very, very clearly always is this activation of double-stranded RNA response. That, of course, is downstream of interferon signaling itself. So it's a positive feedback loop. So what I think is that it's possible that you don't need like a crazy increase of expression of ERVs. You might just need an initial shot, so to say, or maybe some specific repeats that are more efficient to activate or to form double-stranded RNA. I don't know, because this we see in the tumor cells, but we do have more. And with this shot, you will be activated the sensors of double-stranded RNA, so you're basically going to continue maintaining this activated signaling. But it's, I mean, it's complicated bioinformatically. It's very complicated also. So I'm not a bioinformatician myself. It's always difficult.
Okay. Thanks.
So I have also a couple of questions. So if I understood correctly, hypomethylation comes before interferon signaling. I mean, because hypomethylation increases the double-stranded RNA and then the cell responds. So intuitively, it looks like that. Do you know what causes this hypomethylation?
No.
Okay.
Yes, this is,
next question.
No, this is the eternal question, I think, for most of the people in the field. So yes, I mean, this is the idea for us, that you have first hypomethylation and that this is the starting point of the whole thing. Now, if you look to methylation, to DNMTs or either methylating or demethylating enzymes, they're all equally expressed in those tumors. So what we can say is that it's not mediated by this, that they have less DNMT or DNMT mutations, as it happens in leukemia, for example. This is not actually what's happening in solid tumors. Now there are studies pointing to a link to so-called mitotic clock, or like cycling time. So one of the ideas in methylation is that basically when you cycle more, you lose more methylation, because basically you are not able to pass the information of the methylation to your progeny. So if you are cycling more, you're cycling faster, you will be losing methylation more compared to a cell that is doing less. Now, when you analyze these tumor cells, there is a tendency that they have higher expression of cell cycle related genes. So this could be one of the reasons. Normally also hypomethylation is linked to more aggressive tumors and higher grade tumors, was also the case for us.
And regarding the cell of origin, so do ductal cells have less methylation?
No.
Okay.
No, so it's something that happens, I don't know.
Okay. The other question is, so since they have more this hypomethylation, this double-stranded RNA, does the mutation ratio also changes in these cells? Did you check?
I mean, we have few from our study, we have very few samples, so it's, I wouldn't say anything. I mean, pancreatic cancer, you know, is very heterogeneous, so it's always the same core pathways that are mutated, but each patient has a different mutation. So the interferon high, as I mentioned, they associate with this more aggressive subtype, squamous or basal also, and with higher grade tumors, and they tend to be have higher mutation in rate in TP53. So, I mean, and also if you check in TCG data, the ones with mutations in TP53, they have higher expression of STAT1 or interferon. So there seems to be an association there of higher incidence in TP53 mutation, but it's not like, and pancreatic cancer is not, I mean, it's not that some tumors have no and the others have all of them TP53, because also like more than half of the patients already have TP53 mutations. But you do have an association or an enrichment in TP53 mutant tumors.
Okay. And regarding JAK inhibition, do you really think that it would work? Because considering the effect that it would have on T cells,
yes.
do you really think that it could actually?
So the JAK-STAT inhibition has been tried actually, and this is why we chose this compound specifically that I show you. It's called ruxolitinib. We chose it because when we were initially some years ago starting to do this analysis, there was a very nice clinical trial, phase 1 clinical trial of pancreatic cancer, where they used ruxolitinib to treat the patients together with capecitabine. And basically what they found, what they found there is that there were no differences when treating the patients, yes or not, when adding ruxolitinib, yes or not, in the global cohort. Now, when we look specifically to patients that had inflammation, systemic inflammation as measured by CRP levels in the blood, versus those that didn't have systemic inflammation, respond to ruxolitinib. This was I'm saying some years ago. I said, fantastic. It's basically we are saying, because our interferon high, we also think they have and actually we did check in our patients levels also in places that one yes and one not. I don't know if they will try again. I mean, for us it's also the case that it's not again black and white, that all the CRP high and all so systemic inflammation high and systemic inflammation low go in each of the groups. So potentially one could think this is maybe another way to stratify the patients. Maybe you can combine this blocking JAK-STAT first with a shot of interferon later to go into this acute activation. Maybe this would work. I don't know, but this is how in reality is at the moment.
But related to that, do you think that inhibiting the JAK-STAT pathway in tumor cells, like with one hit, is enough to?
No, for this you have to give it constantly. Actually, this drug, the ruxolitinib, has a very shitty half-life. So you have to do it twice a day, and it's not great.
But also, I mean, pancreatic cancers are basically really, I mean, they are not immune desert, but what they are a T-cell desert, right? So they are, I mean.
Well, the immune cold, definitely, yes.
You really need T cells there to be able to go on that. I mean, I really think that perhaps if you would be able to, I mean, I don't know if you can get more viral than your cancers, right? Because in the end you have lots of double-stranded RNA, lots of viral signals, and I don't know if you would awake that with again like a poly-IC, with like a type, like a interferon 3 agonist to try to make it more inflamed. But I mean, it's very difficult.
It could also be that they are T cell. These are because of this IDO-PDL1, 2 that is being expressed. So maybe you bring the T cells back by inhibiting interferon, so JAK, but then the T cells will not be able to respond in any case. So.
But I don't know if you would bring the T cells in. I think also the fibroblasts play a role there, probably. Or if there are some papers showing that actually the fibroblasts block the recruitment from immune cells also. So.
It's mainly that the T cells, they need interferon to respond. So that's what it's scary with this suggested. That's it. I don't know anymore. I mean,
But it's strange.
I think about the textbook. No. Go ahead.
I was going to ask, what happens when you inhibit the interferon signaling pathway in a tumor cells? What happens to antigen presentation by those cells?
I haven't looked to this, but I would assume you have less because basically you bring all these interferon-related genes, and part of this, as Jose was saying before, it's also antigen-presenting molecules. So you would bring this down. I haven't looked into this. I mean, to the tumor cells themselves in vitro, I have to say nothing happens. They are happy and they don't care. But this was the whole thing with interferon signaling, that at the beginning was a bit counterintuitive, that we found that the tumors are more aggressive because it's a signal that first, if as an epithelial cell, if you have it, it's not good. You don't like to have this. It induces apoptosis, and that's why they give demethylated agents to the cells. They found that by activating interferon, the cells at the end, they die because it's a pro-apoptotic signal at the end. But we do definitely find this association with more aggressive tumor cells. And this is why we think, okay, it might be some of the reasons, or additional reasons, there might be, of course, so many others, is this the whole reprogramming of the stroma.
Well, I have one more question. So in all your transcriptomic data, since you have identified these 3 clusters of cells, have you identified any tumor antigen that could be selectively targeted?
No, we didn't look. No, we didn't look at this. But yeah, maybe with special pipeline, I guess one can check. There is a group in DKFZ working on this, but yeah, I mean, they claim that you have T cells in there, but
that would be the therapeutic approach, right? To couple the inhibitor to something that targets only those cells, but not the immune cells.
Yes, I mean, for this,
you can mine your data.
Yes, exactly. For this, we have. Yes, we have, like, I have this other data where I profile the different cell types, no? And this is one of the things that we do, is to check things that are specifically expressed here or there, to specifically look for this, or things that are, like, there are many genes that are actually highly expressed by all tumor cell types. No matter if you're an immune cell, a fibroblast, like, there's a very strong core program that it's induced in all tumor cells, and this, of course, is important when you give target therapy. Stuff like this, yeah.
Very interesting.
Complicated.
Maria, do you have any questions you can ask?
Like many, I don't know if to continue asking or keep it only for Elisa. But are you planning to do some single cell with these tumors given the heterogeneity? That is why. Yeah. There is another one that I'm super curious, why there is this chronic inflammation. So can you group the patients somehow and try to understand, is there a specific habit, or was there something during the evolution, no? During the aging of these people that could have some influence there? So in the sense that I'm really seeing here such a similarity to other chronic diseases and the ones that we are working, such as COPD, and that is a totally different organ, a totally different system, and we see exactly the very same signatures. So it's really impressive.
Yes. So for your first question of the single cell, yes, it's something, I mean, we are not still doing this, but it's something I'm totally in for. Not only at the single cell level, because single cell is complicated when you want to combine many different techniques. So I think you sort of lose also some information when doing single cell, specifically for methylation, for example. You actually lose many CpGs. So I think we would still see the profiles because it's a very global profile of low high methylation, but you also lose a lot of information. No? But yes, it's in the plan, the single cell, combined with what I would really like to do is to combine it with the regional sampling. So not single cell, but actually see
spatial transcriptomics.
Transcriptomic or just sequencing, like also methylome, to see if you sample from different regions of the tumor, how stable the whole thing is. But that's for the future. And then for the inflammation, yes, I know. I mean, I think one of the things and Alvaro, if you are doing more bioinformatics, maybe you can tell also, I think all these, Jose, also all these enrichment pathways that we use come up many times. because when you have some genes, yes, the gene set will come up, right? So one has to look more into detail what genes are there. And yes, of course, if you have inflammation, interferon signaling related genes, I guess, are going to come up. So if you look into COPD or if you look into these inflamed tumors, you are going to come up with the same signatures. For the relation to the patients,
but sorry, Elisa, but your interferon signal is really inherent to your tumors, because even in your immune compromised animals,
yes,
you still have it, and they are really reacting to it.
Yes, this is the, I mean, this was for us when we move into the in vitro with the cell lines to show that actually the cells have this. So it's not something, because it could be the case that we just sample the samples from the patients and they have more or less inflammation, and this is why you see it, but then they lose it, no? They depend from the immune system for this, but we see it in vitro, and this is for us the reason to say, well, there is this double-stranded RNA and this is intrinsic, so they are going to keep this no matter if there are immune cells surrounding or not. But then you have, I think, the whole thing boosts itself, no? Because it's like shooting all the time.
Just a comment. So considering that most of the pancreatic cancers come from pancreatitis, could be this like the origin of the pancreatitis that actually eventually develops on cancer? But maybe checking patients with just pancreatitis maybe would give you the clue for this. So if those patients already have this interferon signaling, maybe if they can be stratified, maybe you can actually predict those that could have the tumor afterwards.
Yes, it's possible. We don't have this information. I mean, I don't know how you can get this information if a patient had beforehand pancreatitis before the
medical history.
Yes, but I mean, you might find some patients, but I don't think, I mean, you don't screen all the, I think that's complicated.
Well,
but
the idea was to find a cohort of pancreatitis patients and try to find traits that afterwards correspond with tumors, and then maybe imply that those could have more risk for cancer.
But this is the case, like the pancreatitis and inflammation, it's chronic inflammation is associated also in pancreas for this. And also in the mouse models, for example, you put KRAS, nothing happens, you need to have inflammation and then the tumor develops. So definitely you need to have this combination. No? In the ductal derived tumors, for example, you need to lose p53 to get tumors. This is also interesting because it also fits with this higher incidence of p53 mutation in our interferon high. But there is not us, but one of our collaborators from the clinics, they are also doing some research, and they have a project where they actually study systemic inflammation of the patient. They have a collection of organoids from the patients, and they basically, their approach is the other way around. No? They start from the serum levels of the patient, inflammation and different markers in the level, and then now go and check in the epithelial cells what differences are there to link the systemic inflammation in the patients to the transcriptomic of the tumor epithelial cells. So one of the pathways they find, of course, is interferon signaling. They find other stuff, and I should read soon more details, but I don't have all the details.
Very nice.
Chicos, it's 7:20.
Just a question, Elisa.
Tell my kids to sleep, so.
Yeah, did you analyze the enhancers, if they are hypermethylated or hypomethylated?
Sorry?
The enhancers, did you analyze or did you check?
Yes, I didn't. I passed this quickly, but when we look in when we do this comparison to the acinar and ductal healthy, because this I didn't say, but this carryover of the cell of origin, it's supposed to be more marked at the enhancers of the cell of origin. So that a bit the theory of the cell of origin from talking to people who are really in the field, like to Iñaki Martin Subero, for example, is that this methylation in the enhancers, and he has nice reviews and papers on this, when you are a healthy cell, this is telling you, this is really controlling the expression of the cell identity genes, right? Now, when you move to cancer, somehow this doesn't happen anymore, because the cancer cells, and this is something very interesting I would like to follow up on also, I think they don't follow the same patterns of regulation of gene expression, right? So you carry over the information of the enhancers but these changes in methylation are not controlling the expression of these genes any longer. And this is also a bit what we see. So when I show you this heat map with those regions that are equally methylated in ductal cells and in our ductal tumors, they are enriched in ductal in regions of the genome annotated as ductal enhancers. So there seems to be something there, yeah.
Okay, so maybe it's time to finish this session. So thank you very much again for the talk and also for the nice discussion.
Thank you.
Thank you for all the participants for attending this first WeCancer seminar. We invite you to, if you're interested in take part, take one of the talks. So you can fill the form that is in the chat or contact us by mail. You are very welcome to have this possibility to present your work. So I don't know,
I still will need at least 1 year to be able to present something.
And also I see some people here that are Spanish, and I think they are not in the CERFA Cancer group. I invite you to join as well. So just ask John and Elisa how to join the group, okay?
¿Hacemos la foto?
Sí, but we need to all put the cameras on, right?
Pablo son mis padres, que mi madre está con la bata. No sé.
¿Qué haga así?
Que se hagan socios de CERFA.
Pon la cámara, da igual, con la bata o con la toalla en la cabeza.
Con el gato ahí.
¿Hago yo la foto?
Yo no sé cómo va.
Bueno, yo es que siempre hago un
Screenshot
de... Venga, va, sonreíd.
3, 2, 1.